Hi, y'all. My name is Nancy Dankwah, and I'm a biomedical engineering student here at the University of North Texas. And I've had the privilege of working under Dr. Elizabeth Skellum on the elucidation of microsporin-like amino acids in fungi. Now, you're probably wondering to yourself, what are microsporin-like amino acids? And they are very small, less than 400 Dalton second metabolite molecules that are commonly found in marine and aquatic organisms that are exposed to heightened levels of UVA and UVB rays. They are also found in their predators that consume these organisms and operate similarly to how melanin operates in animals and human beings and flavonoids in higher plants. Now, they can be found in cyanobacteria, micro and macroalgae, and some fungi. And even though the isolation of these MAAs are common among marine algae and cyanobacteria, such as E. coli in specific, their extraction processes have not really been explored as in depth as they should be with fungi. With the immense amount of MAAs that are potentially undiscovered in nature, there's a propensity that MAAs have therapeutic properties outside of just UVR absorbance. So to further test this, we have chosen 3 fungi, Aspergillus nigelans, Aspergillus clavatus, and Macroportha grizzliae, that have all been identified as MAA-producing organisms based on genomic mining, however, have not been reported to actually produce these molecules in nature. We will use a common fungal heterologous host, Aspergillus oryzae, to further investigate these pathways in depth and to see if these organisms can actually produce these molecules. To actually create MAAs, we found the microsporin groups that contain a modified amino acid linked to the cyclohexanone core that have the specific gene clusters that encode for the segment pathways within our genome for our 3 fungi. We found that the deoxydiglycosyl synthase had been replaced in our fungi with the dehydroquinate synthase, and they also contained an O-methyltransferase, an ATP synthase, and a carbon-nitrogen ligase homolog. Currently, the pathway for MAAs that is used in literature is associated with E. coli, which obviously is not a fungi. And so we are unsure if the xylulose 5-phosphate using translocutarase to create C-heptulose 7-phosphate is correct. However, we do know now that the DDGS, as I said before, the deoxydiglycosyl synthase, have been replaced with a dehydroquinate synthase to create the 2-demethyl-4-gaugesol, which is the main molecule that is responsible for creating our microsporin-like amino acid. After it goes through all of the processes of creating our microsporin-like amino acid, which has an absorbance rate of around 310 to 324 nanometers, we will then use yeast recombination to build our expression plasmids and express this gene in Aspergillus oryzae for MAA production. Furthermore, after these MAAs are created, they can be combined with the cyanobacterial genes, such as the non-ribosomal peptide synthetase, to possibly create the known MAAs in nature, such as schreinerine, which is the highest known absorbent, has the highest known absorbance of 334 nanometers, or microsporin glycine, which has an approximate absorbance of around 310 nanometers. To actually induce this in our fungi, we had seeded Aspergillus clavatus and Aspergillus nigelans on PDA at 28 degrees Celsius and Macroportha grizzliae on PDA as well at 25 degrees Celsius. We had initially seeded these fungi on DPY, but we had found that there had been so-so growth with our fungi, and so we ended up switching it up for the more richer medium. Our plant inoculations were incubated individually for 2 weeks, and our fast inoculations were incubated in triplicate with 3 milliliters of DNA per fungi for 3 weeks as well. We had seen within Aspergillus nigelans and Macroportha grizzliae in specific that in the presence of PDA created melanin, which, as you can see, the nigellans here turned from white, obviously, to the very dark brown colors you can see, and the Macroportha grizzliae turned the actual medium brown, which led us to believe that it's possible that the fungi are creating melanin as a primary pathway defense mechanism against the UVA that was found within the fluorescent light bulbs in the lab. However, we are still going to test to see that with the induction of our MIA producing enzymes, that if they do create these MIAs in the end. So to actually induce the MIAs into our fungi, we performed an array of DNA extraction techniques to collect at least 4 nanograms per microliter of genomic DNA. After successful DNA isolation confirmed through fluorescent detection using a quantum fluorometer, our individual genes are going to be cloned using PCR, and the expression plasmids will then be transformed into our heterologous host to introduce gene clusters furthermore. The only issues that we had during this process was actually getting the genomic DNA to be extracted from our fungi. Due to the very thick plasmid wall of these 3 fungi, there have been actually issues extracting the genomic DNA in high amounts without having any interference with either degradation of the DNA itself or just the inability to actually get the DNA from the fungi amongst the other debris. With the Monarch kit, we found that the DNA was starting to degrade immediately after use. And after about 2 weeks after purification using Aspergillus clavatus and Aspergillus nigilans, we did not use Magnaporthe grizier for this kit. We found that the DNA had completely disappeared. Using the Norgine Biotech kit, we had found that our DNA yield was extremely low, with Magnaporthe grizier being the highest with 0.405 nanograms per microliter. However, we saw later on that we had calibrated the quantum fluorometer a little bit incorrectly, which contributed to the lower yields. However, it still would have brought our Magnaporthe grizier to about to a yield of about 2.73 nanograms per microliter, which was still insufficient for PCR. We ended up settling on using the Sigma Aldrich kit, which since the Magnaporthe grizier and the Norgine kit had the higher yield, we ended up using just that one. And we found that we ended up getting 4.64 nanograms per microliter of DNA, which was finally sufficient enough to use for PCR. So because of that, all of our DNA had then, all of our fungal species had then been extracted using the Sigma Aldrich kit, and they are currently being used in PCR to extend them to about 150 to 100 nanograms per microliter. To actually create the primers that we are using for our PCR, we found the specific DNA sequences that code for our MIAs in literature for each of our fungi. We then analyzed the gene clusters using FGene-SH and then spliced out any of the introns that were located in them so that it didn't interfere with the genetic code that will be used in the yeast recombination phase. The expression plasmids were then built in silico ingenious, placing each of the individual genes between the unique promoters and terminators. So you can see here we have the primer that I created for Aspergillus nigilans. And for our O-methyltransferase, for example, we put it in between the promoters and the terminators for GDP, which is one of the growth factors that is in our plasmid. And so you would see the forward primer, which has 20 base pairs of our gene, is located right here. And our reverse primer has 30 base pairs of our gene, totaling for a total gene pair of 50 base pairs. And each of our fungi had about 2 to 3 genes per that coded for our MAs per fungi. And so any of the orange that you see are the actual genes that I had put in from the literature that we had. So now that we are in the PCR phase, we are hoping that we'll be able to properly extend all of our DNA. And after that, we can actually go into using the recombinant DNA from our yeast or possibly E. coli to grow out our host Aspergillus oryzae so that we can put in these DNA codes for our MAAs and see if it actually creates our MAA that we are looking for. Thank you so much for having me. I am thankful for the Skeleton Department for working with me tirelessly on this project, as well as the McNair Department for giving me all the aid and encouragement to keep going on a project that is a little bit out of my field. Thank you so much, and I hope everyone has a great day.